From Vermont.
Well, thank you. So my topic is particle segregation, and this is segregation of a suspension of particles in an oscillating straining flow. There is a full paper on this coming out this spring in Journal of Fluid Mechanics. So the problem that motivated, rather loosely, this application is a desire to understand how the colon works. This is perhaps why they scheduled me for after lunch here. Most people know that the colon, its basic function is to extract water from the intestinal fluid.
Sorry, can I just stop you? Your microphone's not on.
I'll wait to the Thanks. So the basic function of the colon is to extract water from the intestinal fluid. Your colon is filled with a rather loose suspension of particles and liquid, which is fairly dilute suspension. And the cells of the colon have these osmotic membranes over which they extract the water. But in addition to that, in addition to just being a tube extracting water, the colon has this rather interesting structure where it has these pockets. There's 3 of them going around, and these pockets are called haustra, and they're separated by these little orifice plates. You can kind of see it here, which are called septa. And these haustra, instead of just sitting there, they have this motion that they do rather regularly. About every 30 seconds, your haustral pockets will contract. You don't feel it because it would drive us nuts, but they contract about every 30 seconds, day and night, as we're sitting there. And this contraction frequency of these haustra is not it's not uniform, but it actually varies depending on the viscosity of the fluid that's in your intestines, depending on what you've eaten, and on the chemical composition of the fluid in the intestines. And to regulate these contractions, the body has sensors built into the colon wall, and these sensors connect to a neural network system that is built into the lining of the colon, and that regulates the frequency of the contractions, the phase, the amplitude of the contractions, and all of that. So obviously the body has spent a lot of time and effort evolving this system. It spends energy doing the contractions, and yet when you ask medical people why this happens, and I should say when I started this work, I was on the faculty of the University of Iowa, which has a world-class group in the medical school in GI problems, and many of them were friends of mine. So I went and asked them, why does the colon contract? And they could tell me all these wonderful things about the frequency of contractions and all these physiological aspects that they had no idea why the colon contracts. And if you read the medical literature, the same thing. You find all sorts of diseases that stop the contractions and lead to all sorts of bad conditions for people. But in terms of the fluid mechanics and the particle physics, they have no idea why it does it. So perhaps it's not so mysterious, though, if you think about it a little bit. Certainly this is meant to represent a cross-section of the colon, and these are the water that's being extracted from the cells around the side of the colon. And so this extraction of water would leave a region indicated in green here where the particles are rich, right? There's a rich particle region, and then therefore there's a relatively lower particle region in the middle. And so certainly one reason why this contraction occurs is just to induce mixing, right? It wants to bring this low concentration particle region close to these cells which are extracting water. But I got interested in this because much of my research has of late been related to particle aggregation, and I was interested if, in addition to mixing, maybe this was occurring both to mix but also to induce aggregation of the particles. In other words, by contracting, you would cause the particles to collide and therefore to increase the aggregation. So I wanted to do a little experiment, and this is just a very simple little experiment where I took a box. And I put a suspension of particles in the box, and this is a fairly dilute suspension, about 5% suspension. And I took this box and computationally I squished it one way and it let it stretch the other way, and then I reversed it and kept doing this back and forth, you know, like an accordion. And then I measured the particle collision rate, and I phase averaged that over the contractions. And I developed this cute little theory, which I'm not going to show you, but developed this cute little theory for what the particle collision rate should be. And I wanted to look at the aggregation rate from this. The computations I have here don't have any aggregation in them until later in the talk, but I just wanted to measure the collision rate. So basically we did this with a discrete element method, which has been published in a number of papers, so I don't have to go into it, but it's a fairly standard discrete element method. There's some interesting novel features in here when you have aggregation, but most of our talk, when you have adhesion, but most of our talk we don't have adhesion, so I don't have to go into that. Anyways, what I found was that when I did this, my theory didn't work at all. In fact, what I found was that the phased average collision rate didn't go to an asymptotic value, which I had expected, but it just kept increasing and increasing. And the reason for that is apparent when you look at the particles. So this is a plot which shows the initial particles. The dashed line is the initial position of the box. The solid line that you see here is the position of the box at the current line. So what I'm plotting is a time series. Here's the initial time. Here's what the particles look like after 10 oscillations of the box. And these are fairly small oscillations, about 10% of the total width of the box. And this is after 20 and 40 and 60 or 50 and 60 and so forth. So again, the solid line is the current location of the box. And what you see is that as you do this oscillations, the particles continue to drift inward. And they continue to do this until they reach a point where almost all the particles are touching each other, and they're limited by contact. And if you place y equals 0 at the very center of this box, you can measure the inward drift. So this is a measurement of the root mean square velocity or root mean square position of the particles versus time. And these are results for a number of runs with different Stokes numbers. You're familiar with the Stokes number, the particle relaxation time over the fluid time scale. And so as you increase the Stokes number, this one is about 0.01 and increasing up to about 0.1, so increasing over an order of magnitude. What you see is that this clustering of the particles occurs increasingly rapidly. And so the clustering occurs more and more rapidly. Here they will reach a point where they hit each other, and so their further clustering is limited by the contact of the particles with each other. But you see a little oscillations there. So what happens after a long time? After a long time, these particles will form sort of a lens or a diamond type shape. What it looks like depends on what part of the oscillation you take the picture at, but this diamond type shape will stretch out in one direction and then reverse itself and stretch in the other direction as you have the box in the other orientation and back and forth. So this was not what I expected. Maybe some of you did, but I didn't expect this. So I wanted to understand this a little bit as to why these particles would be doing this. And so I developed a very simple little theory, little one-dimensional theory, because this is a plane straining flow, so the velocity components are decoupled from each other. And it's for an isolated particle, just to get some intuition about this. And basically, I just have the inertia of the particle and the Stokes drag, and the velocity field. This is the fluid velocity field. It's just an oscillating straining flow. And so when you substitute this in and solve for the position of the particle, what you get is this equation, and this being a mathematics institute, we recognize this as the Damp-Mathieu equation. It has two coefficients. This coefficient zeta is the damping coefficient. It's related to 1 over the Stokes number. And this coefficient q is related to amplitude of the oscillation over frequency, again divided by the Stokes number. So the Damp-Mathieu equation, if you recall, has some interesting stability properties. And so let me go to the next slide. So it has a well-known instability that if the parameter q that I showed there, the forcing parameter, gets too high compared to the damping parameter, the equation goes unstable. So if you look in the literature on this, there's some nice papers, one by Gunderson, that gives a sufficient condition for stability of the Damp-Mathieu equation, which I've given here. Essentially, this ratio of q over 2 times the damping parameter squared has to be less than 1/2, and then you're guaranteed that it will be stable. So I did a number of simulations with this simple little model and found that I started off with a fairly small Stokes number, and this S is, if you work it out, proportional to the Stokes number. So the higher the S, the larger the Stokes number. And so what I find is that as you have a small Stokes number, this will oscillate but drift inward. As you increase the Stokes number, it clusters to the nodal point of the straining much more rapidly. And for higher Stokes numbers, this is close to 0.5. There's a little overshoot here. If you continue to increase the Stokes number so that S is bigger than 0.5, you get these oscillations. And the higher the value of S, or the higher the Stokes number, the larger the oscillations. So also this scale is very large, up to 40. So the particles for very high Stokes number are thrown outward far from the center of the box. You can also use this theory to predict what the rate of clustering would be. This is the so-called Fast-Euler approximation, which has been used quite a lot by Professor Balachandra at Illinois and other places. This is for a low Stokes number. You essentially approximate the particle acceleration by the fluid acceleration. And using this, then you can analytically predict what the rate of clustering will be. You find that the phased average particle position varies exponentially in time, and this little coefficient is the clustering rate, which again you find is proportional to the Stokes number. So the higher the Stokes number up to the stability limit, the faster the clustering will be. And if we look at the numerical computations of this Dampf-Mathieu equation, this little approximation falls right on the data, and we can get the coefficient of that. So anyhow, I back to the colon problem. I thought, well, that's kind of interesting. But in a colon, the particles are almost neutrally buoyant, but they have some slightly heavier density. Some of the particles have slightly heavier density than the water that they're suspended in. So what happens when you add gravity to this simple model? So I added gravity, and I find that the drift due to this oscillation is actually capable of suspending the particles against the gravitational field. What happens is that the particles will oscillate at a certain mean elevation relative to the nodal point of the straining, and they'll enter this limit cycle type behavior. This is a plot of position of the particle versus time, and the dashed lines are the fluid forcing, so what a passive fluid particle would do. And the solid lines are what the actual particle will do. And you see there's a phase lag between the forcing and the particles, but the particles will just oscillate at a certain position, a certain distance from the nodal point of the straining. And theoretically, we can use Fourier decomposition to get a solution, analytical solution for what the amplitude of this oscillation is and what the elevation of suspension is. So back to the DEM simulations then, so enough for this simple model. Again, the red squares are the initial position of the box, and the solid is the actual box at the current time. This is a plot where I've taken these particles, put them in a box, done this oscillations for a long time, and looked at the shape that occurs after a long time. These are 5 different simulations with different values of the Froude number. You can define the Froude number in the usual way. And the plots are done at 2 different positions in the phase. So the top is when the box is all the way out that way, and the bottom when it's all the way out the other way. So you can see what this looks like. And so from the theory, you can predict what the critical Froude number for suspension is, right? It's essentially when the elevation height is smaller than the bottom of the box. And so from the theory, we predict the critical Froude number to be 2 for this problem, for these parameters that I've used here. And so if the Froude number is above 2, what you get is that the particles remain suspended in this cluster, even though you have downward gravity. And as you decrease the Froude number, so you're increasing the gravity, you get some of the particles a little bit lower. You get sort of a tail that develops here, but they're still largely suspended above the box. At about the critical Froude number, you see some of the particles start touching the box, and then at very small Froude numbers, they essentially lie in a heap on the bottom of the box. So that was kind of interesting. this maybe I'll skip. It just shows that the granular kinetic energy of the particles as they're oscillating increases the farther the suspension distance is from the nodal point of the straining flow. You can also do this with a mixture of different particle sizes. So we looked at a case where we had these red particles which had a size that were about 3 times larger than the blue particles. The blue particles are the small ones, and initially put them uniformly in the flow, so they were all uniformly distributed, and we did this oscillation for a while. And after about 300 or so oscillations, what we find is structures like this occur where the larger particles having a higher Stokes number will cluster rapidly, and the smaller particles with a smaller Stokes number will cluster and form a coating on the outside of this cluster. So whether this could be useful for forming, I don't know, some sort of self-assembly of structures or not, I don't know, but I thought it was interesting. We also turned on adhesion. So this is with van der Waals adhesion between the particles. Everything I've shown you previously was with no adhesion force between the particles. We looked at very small particles with van der Waals adhesion and found that over time they clustered a little bit more rapidly than the case without adhesion, but they would form these bridges. So after these aggregates form due to the adhesion, you get these bridges between them that would connect, and after a long time, the bridges between the particles would arrest the clustering because the particles would touch through these bridges. So boxes are nice, but I wanted to get a little more closer to the colon. So I thought, okay, well, I can't quite, well, I could simulate a colon, but rather than going to quite that extreme, I thought, well, maybe just peristaltic waves in a tube. So I looked at a case of just a tube with periodic peristaltic waves. So the middle would be going in and out, and the edges go in and out, but the waves aren't propagating. They're just standing waves. And I filled the tube with particles, and I just let them oscillate back and forth for a long time. So you can see here's the time series, and what happens is you get a drift of particles both inward, but also in the x direction, you get a drift of particles toward the nodal points of the straining field. So the nodal point is right at the middle, and there's nodal points at the end. The middle one is the most prominent. So there's a drift of particles towards this nodal point, both in the y direction, but most prominently actually in the x direction. I went back to the literature now and looked at the literature for peristaltic pumping, because there's a lot of literature on peristaltic pumping, and some of it actually has peristaltic pumping with particles in it. And some of the experimental literature actually did notice in passing, usually it was like one line buried somewhere in the results section, that particles actually do drift a little bit inward. I found 2 experimental papers that noticed this and didn't really do anything with it, but just noted this in passing. So this is a case with adhesive particles. So there's van der Waals adhesion between the particles, like you would have in your intestines. Again, with the channels with the periodic peristaltic waves. So there's 3 pictures here. This is what you see after a long time of this occurring. This is for a case where you have a rather small adhesion coefficient, a moderate adhesion coefficient, and a higher adhesion coefficient. And what you see is that, perhaps we'll look at this one, that you get these aggregates forming on the outside near the channels, as well as at the nodal points of straining. And you also get a drift, again, a slight drift towards the nodal points of straining, but that the particles get caught up in the aggregates, so the spacing between the aggregates is actually, larger. You get larger aggregates in here than you would in this part. So I did some looking at the diagnostics of the size of the aggregates and so forth. I don't know how incredibly pertinent that is, but basically this is what you would expect, that the higher the adhesion coefficient for van der Waals adhesion, the larger the aggregates that you get. But this actually a kind of bimodal. You get some, if you look at the black lines here, you get some large aggregates, but you also get some smaller aggregates because the aggregation is most active towards this nodal point of the straining field, and then you get smaller aggregates in the other parts of the flow. Okay, so I wanted to come back and say, does any of this make any sense for the colon? Because this is very interesting and all, but is it relevant? So I tried to do some estimates for the colon, and this is kind of the the typical frequency of oscillations, a length scale, a typical Reynolds number for the colon. And I looked at a different range of particles. In the colon, you have some very small particles. The smallest particles would be about 100 microns. And then you have some larger particles, some of which are aggregates of small particles, and others are more indigestible things that you would eat. For the very small particles, the Stokes number is something like 10 to the fifth. And so the clustering rate is very slow. That you would get from this, and it would take a long time. So that basically tells us that for the very smallest particles, this isn't really a very important thing going on, that mostly they're oscillating back and forth and colliding with each other and probably forming larger aggregates. But this is interesting. As the particle or the aggregate size gets bigger, so looking at about 2 millimeter cases, the Stokes number is about 0.005 or so. The clustering rate is not a huge number, but this is similar to the simulations that I've been showing you. And the clustering time is on the order of a couple of hours, which is typical digestive time would be several weeks. For some of us, shorter for others, depends on how much fiber that you eat. But this is quite compatible with the digestive time that you have going on. So the hypothesis, and I haven't tested this at all and run this off of my medical friends, but the hypothesis that I kind of have brewing is that maybe in these oscillations, the small particles are colliding with each other and forming aggregates, and then as these aggregates get big enough that they have a reasonably large effective Stokes number, they start to drift towards the center, leaving a more dilute region at the edges where the cells are that absorb water. So it's possible, we'll see. I think there's probably some other regions or areas of application where this could be of interest. There's quite a few processes, both biological and engineering processes, where you want to separate particles from a liquid or from a gas. But you don't want to use the typical filtering or centrifuging operations. Maybe the particles are adhesive, so they muck up your filter or your centrifuge, or maybe they're very delicate, like biological cells, where you would break them. So some examples are things like in generation of algae for biofuel production. Separation of the algae from the liquid is a critical issue. And other things such as food production or waste processing, perhaps things like self-assembly of nanostructures we talked about a little bit. There's a number of biological things that we've done a fair amount of work on blood flow using DEM methods. And with blood flow, there's a well-known phenomenon that the red blood cells cluster at the center of the channel. And there's a number of theories for how this happens. And you observe this even with flow through straight channels. But nobody's ever looked at the effect of the blood pulsations, the oscillations on the channel on this clustering. It could have an effect, it could not. So further things to look at. And just a few conclusions, and we found that particles suspended in an oscillating straining flow drift towards the nodal points of the straining, that this drift rate is essentially proportional to the Stokes number up to the stability limit, that this phenomena is capable of suspending particles against gravitational settling, and essentially that we have this simple one-dimensional theory which seems to be reasonably accurate at describing our results. So that's all.
Any questions? Kimberly?
Could you say something about how you simulated your fluid and whether or not the fluid in your simulations responds to the particles, or just as a...
It's one-way coupling. I can put the DEM method up. But no, the fluid is set. It's one-way coupling. It doesn't respond to the particles. I will mention that the periodic straining flow that we used is a solution of both the creeping flow equations and the inviscid equation. So even if there is some modification of the viscosity, this is still a solution of that independent of the viscosity for the oscillating box problem. But certainly, yeah, for some of the other problems, as this cluster forms, you might want to start modifying the fluid flow.
Thank you.
Any other questions? Ur?
The cluster formation is a very strong function of particle size itself. And so you're simulating particles starting off with monosized particle systems in these DEM simulations? Because it's history dependent and it's heavily geometrically regulated. And if you start with a monodispersed assembly, you're going to see a totally different picture than if you start with something which is somewhat polydispersed.
Well, as I've shown you, we've looked at both cases.
Yeah.
We did most of this with monosize just because I wanted to understand the very simplest level what's going on.
Sure.
But we've also looked at two different sizes. We haven't looked at more than that.
Another further comment, alongside the equation sets you use, there's a whole set of equations from percolation theory, which actually could be used to great effect in incorporating the percolation of the fluid through the aggregates, and they show the breakup and the aggregation as being reversible mechanisms at competition with each other. So you're not looking at just growth from very small particles to large ones, but you're also breaking up large ones to small ones. It's a two-way physics that's operating. So it's not just a case of things getting larger and larger still. It's a case of they get to a critical size and they start breaking up, and that's all related to the peristaltic motion or whatever else you're putting into boundaries.
Yeah. In general, I agree with you in aggregate development. It's that balance between breakup and formation. In this case, they do just all form one clump at the middle. So there's not a lot of breakup going on. But
is that true in reality, I wonder?
Well, yeah, so we have to look at reality to see. It's been true in what I've looked at for this mechanism so far.
If you have no theory, you don't know what questions to ask reality. So I think that's very clear. A biological system is notoriously complicated.